So remember I also said that, again, this is review, in the human genome, about half of the genes we don't even know what they do. The only way you can find out, as we talked about on last Thursday and we've talked about many times in the class, it's the only way you can find out what a gene does to have a mutant. It's the only way. It's perturb the gene and see what is lost or what has changed. That's the only way in which you can do that. Now, of course, you're never going to be able to do this in 1,000,000 years in humans for a variety of ethical issues. Some obviously very strong ethical issues. But one can do this in mice. And one does this by germline gene therapy, essentially. Germline gene therapy or germline genetic engineering. One alters the DNA in the cells that give rise to the gametes in the mice and therefore make lines of mice that are mutant for specific genes. And we do this by altering the germline. And the germline are the cells that give rise to the sperm, cells that give rise to the sperm and to the egg. Okay? And how is that done? It's done by some of the ways that you covered in discussion, but it's done in some respects in slightly other ways. And I'm not going to spend a lot of time on this because I just want to focus on it conceptually. And one of the ways is, here's your mouse. You female mouse, you've superovulated it in ways in which Dr. Evans will talk about on Thursday. You get eggs out. You inject your favorite gene into the mouse egg. You implant the egg back in the mother mouse, and you get little pups out. And theoretically, you've added a gene to those particular mice in which they have been born with. So you're genetically engineering the mice. And because you've stuck it in the egg, it's in all the cells of the mouse. And therefore, all the offspring will have it depending upon how it assorts. And you talked a little bit about that in discussion. So egg injection. The other way to do it is a way in which you'll discuss on tomorrow's discussion, but you had a little bit of it last week, and that is, in this particular case, you can take stem cells, and I'm not going to go into that because that's what Daisy and Aicha and Kelly are going to go into, but you can take stem cells that are derived from embryo, embryonic stem cell lines, and you can take those cells and you can engineer these cells in a petri dish with your favorite gene. The gene is taken up by those cells. You can select for them using selectable markers, just like you would be working with bacteria or yeast. And then you can take those cells, inject them back into the embryo of the mouse, take that embryo, inject it back into the mother mouse, and you get little genetically engineered pups out that have the genes in all of their cells. This is all extremely simplistic, of course. And of course, you could also use somatic cells and do the cloning operation that you discussed in discussion last week. So there's a variety of ways in which you can add genes to mice, and you can do this in any mammal. You could do it in humans if you wanted, but there's a million reasons why we don't do this. Now, of course, as Kelly and Aicha and Daisy pointed out in discussion last week, you have to check your mice that are now transgenic for the fact that they have the gene. So you have to do some DNA fingerprinting on the little pups to show that they have what you want. But you can do that, and the gene's going to segregate like a pedigree, but I'm not going to get into that. But the point is that you inject the gene into the egg or into these embryonic stem cell lines, and then you can see whether or not the gene is present in the offspring. You got it, Kathy? It's pretty straightforward. Nancy, Anna, pretty straightforward. It's making transgenic mice. But that's not how you get rid of a gene in a mouse. That's how you start manipulating the mice in some way, but that's not how you manipulate them. In order to get a mutant of a mouse gene, you have to do some other things. And I'm not going to really get into this in any detail whatsoever except conceptually. If you have your favorite gene, Dora, in humans, you can find it in the mouse genome by using a blast search, taking the human gene and comparing it sequence by sequence with the mouse genome, and you'll find your mouse gene and you'll know what it is. You can then take your favorite mouse gene out, because you have a clone of it, and you can work with it in a test tube, so to speak. And the object of working with that mouse gene is you want to insert a piece of DNA inside that mouse gene so that you disrupt it. Just like you're disrupting the beta-galactosidase gene when you do some cloning like you did on your take-home exam. So the point here is that what you want to do is you want to take your target gene, and I'm only going to talk about this conceptually, and you want to add a hunk of DNA to it so that the gene is mutated in a big way. It's knocked out. It won't produce anything. And that's done generally in bacteria. All your cloning is generally done by the procedures which we've discussed in this class in bacteria. And you create a piece of DNA of the mouse that has a hunk of DNA into it, a foreign piece of DNA that now mutates that gene. And then what you want to do is you want to be able to get it back into the mouse, and I'm only talking about this conceptually. You want to get it back into the mouse, and there's the normal counterpart allele. There's the normal one, there's the mutated one, and you basically want to exchange it. In the chromosomes of the mouse, you want to get rid of this, and you want to introduce that. And by getting rid of what was there, Brian, and introducing this, you've now created a mutant mouse gene that won't work normally. Is that clear? Or not? Not going to go into how this is done because it's too complicated. It's just conceptually what you want to do is create a mutant gene. And you create this in exactly the same way you would do, again, introducing a cDNA or introducing anything into the beta-gal gene in the bacteria plasmid vector. You do this all using bacterial cloning. But then what you have to do in order to get it to exchange with the gene in the mouse chromosome, you have to use mice cells. And the cells that you use can be a variety of different cells, but generally they're these cells. These are the mouse cells. You use these embryonic stem cell lines. And they're growing in little petri dishes. They're growing in petri dishes. And you introduce this gene into these cells growing in the petri dish, and they go in by transformation. No different than adding DNA to a bacterial cell. And they go in by transformation, and because of the way you've constructed this thing, and I'm not going to go into why, using the normal processes of the mouse cells, this gene that you've stuck into these cells will line up with its target gene in the mouse genome. And much like you read in your Scientific American for one of your discussions, I believe week 3 or week 4, there's an exchange of information between the gene that you're introducing and the gene in the chromosomes, and it exchanges. And then because you've introduced markers that you can select, you select in this petri dish for these embryonic stem cells that have the mutant gene. You're using all kinds of antibiotic stuff and different kinds of markers. And so the mutant gene is just removing the one that's there by recombination. But you still have these things in the petri dish. But you know it's there because of the markers. You know it's there because of the DNA fingerprinting that you've done. And you know it's there because of everything else. And then you take these cells, and lo and behold, you take them, you stick them inside surrogate mother, you grow the pups, and those pups have the mutant gene. And then you just work with that colony of mice, and you see what the phenotype is. And you see if that mutant gene has any obvious phenotype that would give you some clue as to what the function of the gene is. Enormously powerful. For example, what happens if you take this cell, and you put it back into this mother, and you don't even get any pups back? That might tell you that the gene is very important for embryonic development. And if you don't have it, the embryos won't even develop, or a variety of different kinds of things. Conceptually, this is how you try and find out the functions of human genes using mice as a model by doing all of this genetic engineering from a conceptual point of view. Chris, my man, what's your question?
So, what if there
I know your question's not so.
No, it's not. What if there isn't any obvious phenotype that
Then you're in bad shape, and that happens all the time.
So there's no other alternate way to study it?
There's two problems that lead to a lack of phenotype. The first problem is that that gene might be duplicated in the mouse genome. You might have another copy someplace. So if you want to really find out what and it might be quite related to each other, because sometimes these genes are duplicated, okay? If that's the case, you got to knock out both of them. Well, you know they're there from the BLAST searches. That's not a mystery. You know that from your bioinformatics. So that's easy to do. But then you got to create a double knockout, so to speak, okay? So that's the first thing. Second thing is that, let's say you just don't find any phenotype whatever. You might not see the phenotype because it might not be manifesting itself under the conditions that you grow the mice in your little laboratory. So for example, maybe this gene prevents a pathogen from infecting the mouse or in humans, and you don't know what that pathogen is. So the problem is that unless it's something obvious, you don't know what it's going to be, and if you don't get the phenotype, you're dead. So we can play this game in plants. In our HC 70AL class that I've been advertising, the undergraduates collectively in that class from this class knocked out 100 plant genes using a procedure similar to this. We found 2 phenotypes. It's not so easy, but the point is that it can be done and it is in fact powerful. Okay, Eden.
Is that another way of making a gene library?
Pardon?
Is that another, this process of, I forget, I'm sorry what it's called exactly, but
This is called knocking out genes.
Knocking out genes, is that another way of making a genome
No,
library?
you already have the genome library.
With the
You
bacteria?
already have the genes, yes.
Okay.
So you're knocking these things out in the mouse cell itself. This is in your textbook. It's outlined in very nice conceptual detail in your textbook in dealing with how to study genes from the human genome. Conceptually, it's just called knocking out genes by replacing the gene that's there. Not to belabor the point, you have the clone because you've had the library, Eden, you've had the mouse library. You know the counterpart to the human gene because of the BLAST searches and the DNA sequence similarity. You stick a hunk of DNA in that mouse gene in a bacterial vector by using genetic engineering and restriction enzymes and all of that different stuff. And then you take that mutant gene and you stick it in the mouse cells and you've engineered it so that it's going to replace the copy that's there. And then you grow the mouse back and now it's missing or it has a mutant gene that it didn't have. And then you look at the mouse and you see, does it behave differently? Does it do different things? And it's enormously powerful in terms of observing the phenotype. Okay. Again, probably the most important use of genetic engineering, Dora, in animals is these mouse knockouts. That is looking for genes in humans that have counterparts in mice and trying to find out what they do. It's done in hundreds and hundreds and hundreds and hundreds of labs all over the world. It's done at UCLA all the time. It's done at Davis all the time. Anyone who's interested in human genes, this is their main activity if they want to find out what those genes do. And so more than anything, and I'm emphasizing this, more than the goat, the sheep, the cattle, whatever, mice are the major objects of genetic engineering of mammals, meaning animals. Now, you read last week, and I'm not going to go into it in detail, about also genetic engineering farm animals to make drugs. And you talked about a pig, but people use goats these days and other kinds of, mainly goats, in order to do this. And the procedures for engineering a mouse is the same as a goat. The procedures for genetic engineering a mouse is the same as a human. We don't do it, but it's the same. Procedures for genetic engineering all mammals is the same because they reproduce the same and do exactly the same thing. The procedures for making mouse embryonic stem cell lines like this is exactly the same as making human embryonic stem cell lines. Michelle, there ain't any difference. If you want to clone a mouse, you can clone a human. You want to genetically engineer a mouse, you can genetically engineer a human. You want to do a knockout in a mouse, you can do a knockout in a human. That technology is child's play. any laboratory at UCLA or Davis or anywhere else can do all of this stuff in the goat, in the sheep, in the horse, in the dog, in the mouse, or if someone wanted to, in a human. Of course, you can't do that because of all the obvious reasons. You can technically, but there's a million reasons of not to do it. So the point here is that there's a lot of reasons why to use farm animals for making drugs for human beings, and you went over all of that in discussion on Wednesday. I don't need to reiterate it, but in many respects it's the same reason as for using yeast. Only it's even more powerful in some respects because you only have to manage, let's say, a goat herd or a pig herd as opposed to making big fermenters, which cost a lot of money. Having a goat herd is very cheap. You can make tons of these proteins in the milk of the goat. And all you have to do is use the promoter, the switch, which works in the mammary gland of the goat or of the pig. And as you know from discussion last week, they actually used one from a mouse. And that's because all these mammals are so closely related. The switches of one mammal will work in another mammal. But the point is that you can make any drug that you want for obvious reasons. And you can do it the same way using embryonic stem cells. Or you can, in fact, clone the genetically engineered goat after it's made. Or, alternatively, you can engineer the somatic cells and then insert them into the egg and make a genetically engineered clone. What I'm saying is you can genetically engineer the goat by injecting in the egg, as you know. You can genetically engineer the embryonic stem cells of the goat and engineer it that way. Or you can take the genetically engineered goat or sheep or whatever and clone that by the normal cloning procedures that you'll talk about this week. Talked a little bit about last week. Or you can engineer the somatic cells or the embryonic stem cells and then clone it. You can do a variety of different things using genetic manipulations. This is a revolution in mammalian reproduction, and it's a convergence of mammalian reproduction and genetic engineering. Was that Julie or Eden? Okay. Any questions? I cannot tell you what a revolution this is in terms of what we're able to do genetically. So, you ready? You ready? 1, 2, 3, 4, 5, 6, 7, 8. Stop display. So we have 4%. One brave soul said yes, and most of you said no. What did you say, Dora?
I said no.
Why?
Because they're humans. It's different with a farm animal or something.
Well, that's interesting because when Michelle Evans comes on Thursday, you're going to find out about a lot of parents that get clones of their kids. They're called twins and triplets and quadruplets. Happens all the time. Do you think that if you cloned yourself, it would be Dora?
Yes.
It would?
Kind of.
You ever know a set of identical twins? Are they exactly the same people?
No. But you'll have the same DNA.
Well, that's true to a certain extent. Okay. Who said yes? Who's the brave soul? Albert. Elaborate. In an intellectual way, please. In a way befitting of your intelligence. Go ahead.
I just don't see why it's a problem. Like, if...
Well, give us some reasons. Saying that I don't see it's a problem is not a reason.
I might really like my kids someday and might want one that looks exactly like them.
Okay.
And cloning this kid wouldn't, like...
We're assuming that it's 100% safe now. That they don't have any chance of having any defect any more than doing it the normal way, whatever that is.
Cloning this kid is exactly like having another kid, except for he's going to look the same. Everything about them is going to be completely an individual thing.
Okay.
So I just don't like. If you can choose what you want, why not choose it? It's like going to buy a car. If I can get it in red and I want it in red, why would I take it in blue?
Okay. Who said no? So let's take Mike.
I'd like to raise a specific point that I'm not against it from an ethical standpoint, because people have children all the time, and unfortunately we can't stop them from doing that. I just don't believe that the American public is intelligent enough to understand what they're actually doing with cloning. They'd expect, an exact copy of the child that they missed, or their parent, or even of themselves. They don't understand that it's simply genetic information being transferred. On that ground, if they actually understood that cloning themselves would just create someone that also contained their genetic information, that I wouldn't have a problem with. But cloning parents, children, I feel that gets too far into people expecting.
Well, what do you expect, not what I expect? I don't want you to speak for me as an American. What does Mike speak for himself?
Were I to clone myself, I would expect.
I mean, do you think that, would you say that this is okay or not?
From an ethical standpoint, cloning yourself, I have no problem.
Okay, who else said no? Let's go to Nancy.
Well, I said no because I don't really see why anyone would want a clone. I think it's being selfish.
But that's a different issue,
Yeah.
though. That's a totally different issue. Look at the question and answer it and frame it from that point of view. Yes, maybe it's selfish, maybe it's not. I mean,
Yeah, for the same
would you
reason
say that someone who wanted 18 children is selfish?
Yes, definitely.
But that's your judgment. Now we're making a moral judgment or a value judgment, right?
Yes.
But the other person might want to have 17 kids. So you want to rethink your answer or not? You don't have to. I'm not trying to, maybe you wouldn't do it, but would you want to prevent someone else from doing it?
I think that just because someone has the same DNA, they're going to grow up, they probably will grow up to be different people. So I don't see why people just, if they want kids, why don't they just adopt? So.
Okay, but the question says, would you object to others doing it?
Yes.
Okay, who else said no? Samantha. And then I'll ask another question that I don't have on this slide.
Well, just kind of repeating what Michael said about people grow up differently, and it's not just your genotype who makes who you are, but it's also the environment in which you're raised. And what about these individuals that are actually the clones themselves? Not just talking about the people who are being affected by the clones, but the clone individuals are actual beings and actual humans. And it's kind of hard to have an own sense of identity if you're a clone, and for whatever reasons they came. Obviously, I think it's selfish, but that's not a scientific answer.
Honestly, I've known many identical twins. They don't seem to have a difficulty with their personalities or their identities, quite honestly. Eden, did you say no? Yeah. I'm going to frame it in a different way.
Also, the phrasing "whomever they wanted." If parents wanted to clone Britney Spears for some reason, and they didn't get Britney Spears, then that would be really awkward for the child.
Okay.
I mean, you said whomever they wanted.
Let me ask some questions, and then I'll move on. Does anyone in this room have any children? Amelia, when you decided to have children, did you decide to have children with a specific individual?
No, not exactly.
Okay. But you didn't decide to have children from someone who was random, did you?
No, just from my husband and I.
Yeah, but that means you chose a specific person. That was your husband.
That's right.
That's correct. You just didn't walk around the street and just choose someone, right? But the point here is that you made a choice.
Exactly.
And you made a conscious choice, and you just didn't marry somebody. You married someone special and someone who was dear to you and someone that you had a connection with. And you also married someone of certain features and things that you liked about that person, I'd be willing to bet.
I assume so.
Yeah, exactly. So the point is you made a conscious choice.
Yes.
And out of that conscious choice, you made a conscious choice then to have offspring. Right. And that offspring wasn't exactly random. So let me rephrase this question slightly again. It would be very nice if we would force parents in our society or other societies to get licenses before they had children just like we get people to have driver's license, because it would end a lot of misery in this world, as all of you know, but we don't do it that way. It's harder to get a driver's license than to have a kid. But the point here is that let's go back and take this question and say, should there be a law that would prevent someone from cloning themselves, their children, their parents, or anybody they want? Should this be legislated? Because the question says, I would not object, but now let's say there should be a law that would prevent somebody from doing this in legislation. And so that's the question. So why don't we answer that from that point of view? Now we're encoding it.
Should there be a law is yes?
Yes. Should there be a law is yes. Should there not be a law is no. Okay. So now we have 75% of you say that there should be a law to prevent cloning. That's very interesting. Of course, I hope you realize that that violates the 14th Amendment of the Constitution. Hope you realize that that violates the right to privacy as established by Roe versus Wade. In other words, that the government is not going to get involved in reproductive processes of individuals. The point here is we're going to start discussing that next week or more. You're treading on some very sensitive grounds here. Because if you can legislate that people can't clone themselves, which you could. I mean, we can legislate anything we want. In reality, where do you stop and where do you go? Does that mean you're going to legislate against having therapeutic clones that you're going to talk about in discussion tomorrow? That is, cloning yourself to be able to make a pancreas to keep yourself alive? Cloning your child? Hold your hand, Chris, I see it. Holding your child, cloning your child in terms of just an embryo, not a human being, but a little embryo so that they can get bone marrow cells in order to be able to keep them alive. Without that, you would die. I mean, this isn't black or white here. And so, where does it stop if you want to pass a law to prevent someone from cloning themselves? Where does that stop in terms of, well, now you can't do this, and now you can't do that, and now you can't do this? One of the most wonderful things about genetic engineering, there hasn't been any laws passed that say what we can do and what we can't do, because it becomes your precepts or morals or desires may be different than mine and somebody else. And when it comes to reproduction, it starts treading on some very sensitive grounds here. And so we're going to pick this issue up again next week. But I just want to give you a preview because it might not be as simple as a yes or no answer, particularly as it pertains to a variety of other things. Should we legislate that two males who want to have a child shouldn't be able to have a child? Should we legislate that two females who want to have a child shouldn't have a child? Should we legislate that one can't use donor sperm or donor eggs? Should we legislate that someone shouldn't be able to use a surrogate egg or a surrogate in order to have a child? I mean, where do you stop in terms of reproduction? And so I don't have an answer for this. You have to answer this because different societies have different ways of looking at it. And if you read your packet for discussion tomorrow, you'll see that different societies have different ways of viewing stem cells and what you do with stem cells, because it was wonderfully written in that packet. The question is, where do you go with this technology and where does the bar raise? As a footnote on that, it's only been since 1972 that in vitro fertilization has been taking place. And in the beginning it was so controversial, and now it's done all the time. And so things change. And I would venture to say that with this technology, as it becomes more and more and more and more and more and more and more perfected, that the bar will change as well. And so I want you to really think about that in a critical way, and we'll come back to this at another point. Okay, so we've talked about making recombinant human proteins in animals. I'm not going to get into this in any... Who was that? Oh, I don't want to get into this in any deep way. We've gone into that. I just want to say that one of the reasons why one would never want to do this in humans is because it's a very inefficient process. And you can see right here that the transgenic progeny, which are born as compared to the control, whether it's cattle or pigs or sheep or mice, is only a very small percentage. It's an enormously imperfect process. And that's exactly one of the reasons, among many, that one wouldn't want to touch this with humans with a 10-foot pole. And it's very inefficient because of what you're doing and how you're doing it. But it's also inefficient because maybe when you inject a gene or something else, it's mutating another gene. We've talked about that. And so the point is that in order to get a transgenic goat to make a drug, you probably have to do this 200 or 300 times. So it's enormously inefficient. And so it's okay for mice, it's okay for pigs, it's okay for goats. And here's the first genetically engineered goat that's been approved for a drug. And it happens to be a drug that makes a very, very interesting drug, actually, as I'll get into in a second. This is the goat that's been engineered with this thing called atrin, and it's antithrombin deficiency disease that its target is. And antithrombin deficiency disease is involved in this coagulation pathway. And imagine that there has to be something in your blood that would prevent clotting from occurring because these proteins naturally occur in your blood. And so you have this thing called antithrombin, and this antithrombin prevents the interaction of thrombin and fibrinogen under conditions of which a wound would cause a clot. And people that don't have this gene, this antithrombin 3 gene, which is a genetic disease, people who don't have it spontaneously form clots. And in order to prevent them from having clots, they have to take certain drugs. And by taking certain drugs, they don't spontaneously form clots. And it's really a problem with pregnant women in terms of mortality rates and other kinds of things as well. So, and you're susceptible to heart disease and all kinds of stuff. And so this antithrombin deficiency disease, now that you're producing this antithrombin human equivalent that can prevent clotting by just taking it like you would, factor VIII or something else, is a major breakthrough. And it has to be done, at least in eukaryotes, because of the modifications of these proteins, which we've been talking about, that occur after translation. And so these goats, which have been approved by the FDA, been approved for how they're treated, how they're grown, how the gene was put in them, how the drug comes out of their milk, how it's purified, and been tested through clinical trials, stage 3, is something which I think is a major, major, major step in terms of doing things. Obviously, there's other animals that have been genetically engineered. Every animal that you can ever think of under the sun has been engineered with a particular gene of one type or another. Here is a genetically engineered salmon. In reality, this picture is kind of deceiving. These little salmon will get to this size. They just get to this size slower than these salmon. They don't get to be these giant Godzilla salmon. They get to be the same size as these salmon. They just do it a little bit faster. So in some respects, this is a little bit deceptive. It's not like the super mouse. You're not going to see this in the grocery store anywhere soon for a variety of reasons, perhaps pushback by consumers, environmental groups. However, there's a lot of myths here that I'm not going to go into that are perpetuated about these transgenic salmon. And here are all the responses to these myths from a scientific point of view. All these transgenic salmon are sterile, for example. They're not going to mate with the other salmon. They don't out-eat the other salmon. It's a variety of things that are out there that make these salmon actually pretty good in terms of what you might want to do about them. But now I'm going to come back to a slide in which I showed you a few weeks ago. And in reality, what you're seeing up there, Marvin, is a very important slide. Because if you have a genetically engineered goat, if you have a genetically engineered salmon, there's a variety of issues, containment issues, variety of other issues, but clearly, in dealing with those issues, one has to deal with these issues. It has to deal with these issues. the basic scientific issues in dealing with anything regarding a transgenic organism. You have to deal with, is it a gene? Is it working in the proper place? Is it going to go haywire? Is it going to create a monster? Obviously not. In other words, when you're trying to counteract non-scientific issues, you have to say, oh, the giant salmon are going to take over the ecology of some place. Well, that's the question. If that's the question, that's your hypothesis. If that's your hypothesis, you have a prediction. If you have a prediction, you can test it. You can at least test it in controlled conditions. In other words, there are solutions in order to deal with these things, and people who think about making these organisms think about how to mitigate any potential problems. But it all goes back to science-based solutions. And if you remember Alan McEwen, it's not opinions, it's not a poll, it's not what people think. It has to do with very critical scientific issues. And I would hope that you guys would be able to come up with these kinds of reasons in order to counteract, and I know you have in discussion, and I've heard some elegant discussions of how that works. Mike's had some really elegant arguments, both for and against some of this technology in his discussions. You're a good actor, Mike. It's a good calling. I don't know if you're going into that, but you'd be pretty good at it. Okay, let's turn to plants. And I'm not going to spend a lot of time on plants, but more time than you probably would have thought about. Probably one of the biggest uses of genetic engineering on the face of this earth, by far, in terms of impacting the most land area and the most people, is genetic engineering of plants. Because all of you, I'm sure, unless you're just outright organic eaters, most of you are eating genetically engineered foods all the time, whether derived from soybeans, which are in the United States about 100% genetically engineered, or corn, which is about 50% genetically engineered, or if you wear cotton shirts or clothes, it's 100% genetically engineered, or cotton oil, or any kind of things that derive from very common plants, those things are genetically engineered. So the point here is more acres across the world that I'll come to, more people, literally hundreds and hundreds of millions of people have come in contact with these genetically engineered organisms. And again, from the point of view of genetic engineering, this technology probably impacts via plants more people on the face of this earth collectively than any other form of genetic engineering. Here are all the things that have been done in laboratories around the world using genetic engineering in plants. Plants that are insect resistant, as you know, and I'll show you some, viral resistant, herbicide resistant, fungal resistant, as we talked about here. Plants that are, as I'll show you, cold resistant, drought resistant, salt resistant, as I'll show you, plants that have delayed fruit ripening, plants that have altered nutritional qualities, for example, decaffeinated coffee, allergen-free peanuts, allergen-free soybeans. We have genetically engineered plants that make plastics, genetically engineered plants that make drugs, genetically engineered plants that make vaccines. Almost anything you can imagine has made in any laboratory around the world in using genetic engineering in plants. It's really a very powerful, powerful technology. Only a few have ever gone to market, and those few are virus resistance, herbicide resistance, bacterial resistance. Those are the major ones that have gone to market, meaning have been approved by the USDA, been approved by the EPA, and are out there and have been out there for about a decade. Many of the other ones have not gone to market for reasons that we'll get into in a second. I'm not going to go into this. You did this a little bit in discussion, but the point is that there are literally 300 million acres, 300 million acres of genetically engineered crops grown around the world. It is the most rapidly adopted form of genetic engineering that's ever been done. Farmers love this technology, as Alan McEwen brought out, and perhaps you brought out in your discussion. But the point is, it's been rapidly adopted, more rapidly than almost any scientific advancement in the history of agriculture. and it's grown in many countries around the world, mostly in the US and in China, but it's increasing tremendously and increasing every single solitary year. Most of the traits are herbicide resistant, insect resistance, or both, and there's going to be a whole bunch of others, and it's mostly soybean, corn, cotton, and canola. And I won't get into specific stories on that. Most of the advances, as you know from your discussion, in terms of helping people, in terms of economic advancement, has really been to poor people. In other words, if you're a poor farmer in India or in the Philippines or in China, and I could document this with films, but I'm not going to do it, and you don't have to buy insecticides anymore, and you can use a genetically engineered insect-resistant plant, as we've discussed. You now don't have to buy the insecticides. You don't have to have your wife and children going around in a sprayer in the field, as we've talked about many times in the class. And you get a better harvest, just like I showed you in that time-lapse photography of insect-resistant genetically engineered tobacco plants. So your income goes up. And in fact, the income is almost doubled in many places. And those are United Nations figures. Now, I'm just going to give you examples. Just going to take you through just a few examples. You know the technology. You know the particle gun. You know the agrobacterium. I'm not going to do that. But the biggest loss, one of the biggest losses of crop yield, that means food supply. Maura, do you understand that? Crop yield means food supply. If you're in Africa and you get infected with these pests, you lose your crop, it's not simply that you're not going to be able not to go and buy a game for your kids at GameStop, which they don't have. It means that you're not going to be able to feed your kids. So it's a serious business. And so one wants to engineer for pest resistance. And that's been done many times. Here's fungi, here's viruses, here's nematodes, and here's insects. How many of you have ever read the Bible? You know about Moses trying to get his children, you know, from the pharaohs? And you know about the locusts that were brought to bear on the Pharaoh for going after the Israelites? Well, that's going all the way back, okay, in terms of insects affecting crops and all kinds of things. How many of you ever seen pictures of locusts going across the land? They pick it clean. Clean. There's nothing left. So it's serious business in terms of agriculture and foods. You know how to make an insect-resistant plant. I'm not going to get into this, but clearly that's an important technology. What you don't know about is, how many of you ever been to Hawaii? Can I see your hands? How many of you ever eaten papayas? Well, if you're eating papayas from Hawaii, they're genetically engineered. Because viral-resistant, genetically engineered papayas, these on your right and these on the left are not genetically engineered. Viruses have almost wiped out, or did almost wipe out, the papaya industry in Hawaii. And by genetically engineering for viral resistance, and I'm not going to say how, but using viral resistance genes, it reestablished a wonderful papaya industry in Hawaii. So engineering for papaya resistance, viral resistance. How many of you ever heard of, is anyone in the room Irish? You know about the Irish potato famine. Well, many Irish are in the United States because of that potato famine that killed millions of people. And it's all due to the fact that there was a certain pathogen that happened to be a fungus that would kill a potato plant. And now you can see that we can have fungal resistant, by using genetic engineering, potato plants that you can put all the fungus you want on them and they're not going to do anything because they've been engineered for a resistance gene. That's the power of genetic engineering in plants and in agriculture. How about abiotic stress? Probably the other major cause of yield reduction, that is loss of food, is drought, flood, heat, high salt in the soil. Try growing plants out in the Mojave Desert. It ain't going to work. Poorly irrigated soil. It's huge losses of plants and crops. So one would like to engineer crops to be resistant to drought, to be resistant to cold, to be resistant to a variety of these abiotic stresses. So notice a transgenic plant over here, a transgenic plant over here. This plant here, the one on your right, went a very long time without being watered. Look at the control. Looks like one of my plants in my house. Look at the genetically engineered plant. Doesn't use a lot of water. That's a big deal for farmers. As water becomes a very scarce resource, drought resistant is probably one of the most important traits that we have to get into crops, to be able to use a lot less water. And I can assure you, the folks up where John lives, and Ryan and Dora and Faria lives up in the Sacramento area, know very much what it means about drought resistance and water resources and managing water. It's a big deal in California. And so doing something like this would be important. And of course, cold resistance. Here's a plant that's been treated by freezing. This genetically engineered plant, not even touched. And you know, how many of you know about citrus growers where the freeze comes, they lose their whole crop? If you could engineer crops with these kinds of things and other kinds of crops, you're going to again cause an increase in yield. Increase in yield means more food, et cetera, et cetera. Salt tolerance. Here's a plant that's grown, engineered plant with a lot of salt. Here's one that doesn't grow at all, that's not genetically engineered. So by all of the genome projects that are going on in plants, and by isolating all of these genes in labs all over the world, and I'd venture to say that there's probably more genes being isolated all over the world that are involved in all of these important traits of agriculture than anything you can imagine. And by putting them inside plants, look at what you can do. Salt resistance, herbicide, salt resistance, drought resistance, freezing resistance, fungal resistance, bacterial resistance, insect resistance, viral resistance, all kinds of things. What about nutritional quality? Well, you've all heard about golden rice, and I'm just going to say that this is a rice which is fortified with vitamin A. Literally tens and tens and tens of thousands of kids and people across the world die or become blind due to vitamin A deficiency. And so one has been able to engineer rice seeds with a daffodil gene and a bacterial gene, constructing a new pathway in the seed of the rice, a pathway that makes vitamin A where the rice seed couldn't make vitamin A. It's a very elegant example of genetic engineering. Enough to make so that people can eat the rice and get enough vitamin A in order to prevent this very, very, very horrible disease. It's taken 10 years and it's still not on the market yet. And I'm going to get into that in a second. But the point here is that this technology can help agriculture, can help people. Richard Hamilton talked about biofuels. I'm not going to talk about it very much, except one has to engineer the plant in order to make more biomass so that you can collect more biomass in order to make more biofuel. And this is nothing new. Remember how all plants were engineered by our ancestors thousands of years ago. Here's a foxtail millet that was domesticated by our bioengineering relatives. Here's the wild type variety. Here's a gene that we can put into a model plant, and notice when you put that gene in, the plant gets bigger and the control is smaller. So one can engineer plants to make more biomass, to make them larger with one gene or a few. So the genes that were responsible for our ancestors doing this, we now know what they are. We've isolated them. We've cloned them. We can do this. There's no mysteries anymore in terms of what we're working with. How many of you heard about codeine? How many of you heard about nicotine? How many of you heard about caffeine? How many of you heard about cocaine, cocoa, aspirin, all from plants? Have any of you ever seen a willow tree? Aspirin comes from the bark of the willow tree. Probably never knew that. It's a salicylate. The willow tree is Salix babylonica. The point here is that drugs traditionally are from plants before we were able to synthesize them and go through the miracle of what we're doing today. So one can use the plant just like the goat in exactly the same way for exactly the same reason. Why not take the tobacco farmers in South Carolina and get them to grow tobacco plants that make healthy human drugs rather than growing tobacco plant that makes nicotine that is addictive and can kill people? The point here is that we can engineer plants. In this particular case, we have a plant that's engineered with a heavy chain and a plant that's engineered with a light chain. You put them together into the hybrid, and you're making an antibody. And as I'll show you, you can do this in plants, and you can make antibodies to specific cancers. And you can make these in plants because it's cheap, easily done, and the plant modifies the protein just like the goat does, just like the yeast does, because it's a eukaryote. So there's many reasons to be able to do this. It's stable. You can do all kinds of stuff. So you can make essentially vaccines in plants. You can make all kinds of drugs in plants in a variety of different ways, and that's called, of course, farming, just like with the goat, for a variety of reasons. Now, let's say you're living out in Africa. You know how many children die every year to diarrheal diseases? Bacterial, viral, diarrheal diseases? We're talking about 5 million children every single solitary year in a curable disease. Do you think, Lauren, that you're going to haul syringes to, Malawi of some vaccine that has to be, really stabilized and needs refrigeration and needs to be transported? You think you're going to be able to do that? Exactly. At a cost at which those people can afford? But what if you could make a vaccine in the leaf of a plant, and what if you could just grind up the powder and put it in a little capsule for about a penny a dose and have someone eat it and take it as an oral vaccine, infinitely stable, cheap, transported all over the world and stable? And what if you could make this vaccine that would combat every pathological bioweapon organism potential, and you can have all these little pills in soldiers to be able to protect them against these bioweapons? Well, you can do that in plants. And you can do that in plants cheaper than you could ever do it in goats or anything else, because it's infinitely stable, the proteins that are made. And so the point here is that one can make vaccines that you can either eat to combat a number of diseases. Vaccines have been made in plants for anything for AIDS. It doesn't work very well, but it's been made. But certainly other kinds of vaccines have been made. Hepatitis vaccines have been made that do work in plants, and they're made because plants are really good bioreactors, and it can be done relatively cheaply, and they're stable, and they can be kept around for a long period of time. And so here's some recent news. We have a cancer vaccine made in tobacco plants, just recently released and approved by the FDA. And so therefore, this is a vaccine which we talked about last week, which can combat particular cancers and a variety of things. Now, despite all of this, however, genetic engineering has not been universally welcomed. There is a small but vocal group of people, primarily in Europe, who are opposed to the use of genetically modified crops. So the point here is, that was my voice, the point here is that there's been a lot of opposition to this technology. There hasn't been an opposition to genetic engineering technology for making cancer drugs. There hasn't been an opposition to genetic engineering technology to make better stem cell lines. There hasn't been any opposition to genetic engineering technology to make drugs that would enhance health or longevity of humans or animals. But for some strange reason, people have been, mainly in Europe, against genetic engineering for a variety of reasons. And I know you talked a little bit about this in discussion. I must say that I've spent the latter part of about 3 or 4 years of my life dealing with this issue, because that's what we work with in my lab. The point here is that if you go back to when this technology was invented, which was now about 28 years ago, and you look at all of these different reports of the National Academy of Sciences and other agencies here and around the world, as Alan McEwen pointed out, because he was on many of those commissions, The unanimous verdict of every serious scientist that there ever could be is that there's no harm in any of this technology, no matter what. And if there are potential side effects, environmental issues, those things can in fact be mitigated. In other words, we don't throw out antibiotics because we get antibiotic resistance. We mitigate it. We don't throw out a whole science because someone has an opinion that they don't like it. The point here is that I've just shown you how much good it can do for humanity. Now, why has there been so much opposition to this technology? Let me spend a couple seconds and give you a few reasons for it. Again, it's mainly been based in Europe. There's a variety of reasons for that. People in the US, in China, other parts of the world a little bit more pragmatic. But the point here is that first thing is really been just ideological anti-tech. It's always people that have been around, as you know from taking this class, that didn't even want genetic engineering to move forward in 1973. Some people just think that they just don't like the technology. It's not natural, whatever that is, like these lights are natural, like the air conditioning in this room is natural. They just don't want it. Second reason is that they don't trust their institutions. And I can really understand that. Look at what we're dealing with the salmonella in peanuts. Our USDA really has fallen down. Our FDA has fallen down. They should have caught that. But in Europe, they had dioxin in their Coca-Cola. They had mad cow disease that killed people in their meat. They don't have as strong institutions that protect their food supply. And so they had a lack of confidence in government. They said, I don't want to touch it because I don't understand it. I don't know what's going on. And that means another reason is because, down here someplace, is that they have a lack of public awareness on the science-based thing, which is why I showed that slide previously. There's another reason that has nothing to do with the technology, has nothing to do with not understanding science, has nothing to do with the EPA. It simply has to do with, again, anti-globalization. It's been a merger of a variety of forces. People say these American corporations are going to take over the food supply. It's absurd, but the point here is that it's an anti-market, anti-globalization force. Other issues are in Europe. Look what happened with the Nazis doing eugenic experiments, thinking about, again, as we've discussed in here, pure race, eugenics, all these things. Very wary of genetic manipulation, as they should be from their experiences. Protectionism, etc., all kinds of issues. But one of the biggest one is organic growers, which are big in Europe. And as I've said in a few times in different fora, organic growers have a certain way of farming, and genetic engineering is not permitted along the getting certification, let's say, for organic produce. And they're one of the major oppositions to genetic engineering, so it's an economic issue. But there's a variety of different issues. All of these issues, of course, have been taken up by these commissions here by serious scientists lately. Again, the National Academy of Sciences have shown that there's no issues with these things. So most of the opposition is non-science based. It's more ideologically based, more economically based, more trade competitive based, or more from a livelihood point of view as compared to a scientific point of view. Has it affected real people? Absolutely. As maybe Alan pointed out, I'll remind you, or I will tell you, that in many respects, many African countries prevented good food from going to their starving people because they were worried that their food supply would be contaminated. In some parts of Africa, their leaders said it was poison. They would poison them. You know that all of these things are absurd, but in fact, it did hurt real people. I made a documentary for television, which took me 2.5 years. I'm not going to show it to you. If any of you want it, I can give you a DVD on this topic. When I was in India, I went to the CARE office. I tried to interview these people about using genetically modified foods in India. They wouldn't even touch me with a 10-foot pole. But on the side, they said that if we were seen interviewing you, the activists would essentially picket us and shut us down. The point here is that it has had an effect on real people by not providing them, one, the technology in which they can raise their livelihoods, two, food to keep them alive, and a variety of different things on the basis of choice. And so let me show this to you, if I can.
See, I've got no problem if you decide to live in the past and if you want to deny yourself the potential and possibilities that technological change brings about. That is your business. That is your affair. What I'm concerned with is if you burden the people of Latin America, Asia, and Africa with your prejudice. Most people in this globe are still relatively poor. Most people are still having to struggle to survive. Unless we find a solution to the problems of everyday life that they face in Africa or in Asia or Latin America, they aren't going to improve the quality of their life. And if they're going to be poor, the whole of humanity is going to be impoverished by that process. So in that sense, anti-scientific thought is not just simply about science. Anti-scientific thought is ultimately anti-people.
He's a really good guy, professor of philosophy in England. But the point is that what he's saying is absolutely true. If you're going to eliminate all avenues of science which can be used, as you saw in the Bronowski film I showed you a week ago on the ascent of man, most of us who are doing this stuff, Dr. Harada, myself, and others, we put in long hours. And we don't do it because we're making $5 million a year. We do it because somewhere in the back of our head there's a belief that maybe we're going to find something, and by finding something it's going to help people, maybe one person, two people, or many people, for the good of humanity. And so when you shut down a whole science for lack of scientific reasons, in some respects it becomes a human tragedy. And I would hope that all of you have learned enough this quarter to be able to reason in a very vigorous way, regardless of what this technology or any other technology is being used for. And that is the march of progress really shouldn't be stopped, because you yourselves are the fruits of that march of progress. You use computers, you use air conditioning, you go to beautiful buildings, you have state-of-the-art stuff that we use in the classroom, and you're the beneficiary of all of this science and technology, which in fact has altered our lives more than anything on the face of this earth. And I doubt very much whether most of us would want to go back to live the way the Neanderthals lived. Okay. Let me finish up this discussion on genetic engineering, and I'm not going to be able to finish it today, but I'm certainly going to finish it on next Tuesday before I start the law, and that is that the pinnacle of genetic engineering, obviously, is being able to genetically engineer human beings. And as I told you, it's kind of remarkable to me that people would picket all over the world and spend time and energy against genetically engineered plants when there's genetically engineered human beings that are walking around on the face of this earth. And no one seems to ever have been in opposition of that, because those human beings are alive because of the development of this technology, and their parents would have never been able to enjoy, and they wouldn't have been able to enjoy their wonderful lives, they're now teenagers, without having a gene in them that they weren't born with. And so the point here is that this technology not only has all the applications that I've told you about, but has some very powerful applications with respect to humans. Not simply basic knowledge, which is the biggest application, understanding who we are, what our cells are, where we come from, what our cells do, what the proteins are, and being able to intercede in these processes, but being able to treat and correct genetic diseases. How many of you know someone who had a Tay-Sachs child that was a beautiful child and died instantaneously at age 4 or 5? Can you imagine how devastating that has to be to a parent? How many of you know of a person or a couple or an individual that had a child that was born with an incurable genetic disease, in which perhaps this technology could have corrected that disorder and prevented that particular tragedy to happen? This is powerful stuff, using this technology. Powerful in helping individuals and helping families and helping people. But it has a lot of ethical issues associated with it, which we'll discuss. There's two kinds of gene therapy, as you know, relative to humans or any mammal. The first is the germline gene therapy, which I talked about in the mouse, which I talked about in the goat, or the cattle, or the sheep, or the pig. The gene goes in the germline that makes the sperm and the egg, and that gene is perpetuated as long as that animal breeds and exists, and to their offspring. The second kind of gene therapy is called somatic cell gene therapy. And that is you just engineer cells that don't give rise to gametes, somatic cells. That means you can engineer those cells, maybe put them back, not maybe, put them back in an individual, but they don't become part of the cells that form sperm and egg, so they're not passed on to any offspring. The engineering dies with the life of the individual. So that's two forms. And those are the forms that can take place with respect to human genetic engineering. The first form of germline gene therapy no one would ever attempt for obvious reasons I've just shown you. In the mouse and the goat, it's inefficient. It's unethical. One could maybe mutate a human gene that could make a human being worse than what you're trying to correct. It's not something that anyone would ever attempt to do. On the other hand, somatic cell gene therapy is one of the hottest areas of genetic engineering with humans, has been done, and in fact has worked. And that's the gene therapy which I'm going to focus on for the most part and show you how it's been used to correct cystic fibrosis, to correct the bubble boy disease, SCID, severe combined immunodeficiency, and a variety of other things.